Bene, buonasera, bentornati e tornate su MeetScience. E questa sera abbiamo una grande e importante novità. Ivano ed io andremo ad intervistare un ospite, Akhil, di cui siamo molto felici, nel senso che siamo molto onorati della sua presenza e lo intervisteremo in inglese perché e quindi questa sarà la prima volta che andremo a fare un'intervista di MeetScience in inglese. Speriamo che questa cosa vi piacerà e soprattutto vi invitiamo a seguire la puntata di oggi per una serie di motivi, tra cui l'interdisciplinarietà, la challenge dell'inglese, questa è una cosa interessante sempre, e poi ecco, la curiosità che a me quantomeno ha ispirato il tema di cui andremo a parlare, che è particolarmente interessante, oltre che di frontiera di ricerca. Non a caso ne parleremo, ma insomma anche l'ha appena pubblicato molto bene, quindi ne parleremo presto. So I will switch now to English after this quick introduction for our Italian speaking audience. And I'm super happy to have you here, Akhil. Very welcome on MeetScience. And I hope that you will enjoy this experience. And yeah, first I have to introduce you a little bit at least. And I will be happy after this if you could say 2 words less formal on your education and where and from which path you come from and how you ended up doing research in such a niche and anyway very frontier and interesting field of science, which is actually it was new to me in the first place, but it was like a wow effect because I didn't know that such a thing that you do was possible even. So Akhil Jain, is a research fellow at the University of Nottingham, and after first education years and research in Iran. And Akhil's research now focuses on developing new technologies to modulate cell function. So it's an application in biology of technologies that arise, that stem from the applications of principles of nanotechnology, surface chemistry, bioelectronics, all integrated together. So we are speaking about quantum medicine tonight as for the title of this interview, because we are going to speak on how the physical properties of matter can be exploited for biotechnological applications, especially with nanoparticles and nano-based approaches. Ivano, I don't know if you want to add something in the beginning.
Well, not really, but I am very excited actually to hear about this, because I mean this is the more applied side of the story about quantum mechanics. I deal more on the about the abstract side of things. So I'm very excited to hear about the marvelous things that can be done with this technology.
Okay.
So we're very happy to have you here.
Thank you. Thank you for having me. And thanks, Ivano and Julia, for introducing me. So I'm from India. I'm born and brought up in India. And I did my undergraduate and master's in India in biotechnology. Then I moved to America to do PhD in material science. And then I came to the UK to pursue postdoc or research fellowship in bioelectrochemistry. So as you mentioned, quantum, we had no idea. It's new to me as well. We just discovered this quantum thing last year. We had no idea what is quantum physics, quantum biology, no idea about it until unless last year. That's the beauty of science. Until you discover it, you have no idea what it is. So yeah, it's not new to you guys, it's new to me as well. But I'll try to based on my knowledge, I'll try to educate the audience to tell about what is quantum biology and how we can use quantum medicine.
Yeah. And yeah, and I wanted to ask you if you ever expected to find, this is a personal question. I mean, it was something that I wasn't thinking about before hearing you speak now. But did you ever expect when you started your career that such a field as exploiting quantum properties of macromolecules that you could really manipulate as an instrument might have been something that was going to be a field of research in during our career time lapse? Because to me it was wow. I mean, I didn't know. I could have expected some years ago when I started studying to have some advances, but little ones, not really so big that made us able to do such application we're going to talk about tonight.
So to be honest, I had no idea about quantum biology or this can exist as a new field of research until this COVID happened. So during the part of my research, it was an unexpected discovery, to be honest. We reached to a tight end where we didn't realize what is happening in the biological system, what we see. And COVID actually came out good for us because we got that COVID lockdown break to read about other people's work. Watch some documentary. We had Jim Al-Khalili, who is basically the new pioneer of quantum biology and the quantum physics. We talked to him, and then it came the idea of quantum biology. Then I started to study about what has happened before, like in quantum biology world, starting from photosynthesis, DNA repair, and everything. That time I was exactly like I was shocked, like wow, this new field, because as a biologist, for me, quantum in biology cannot occur at all. Like it's so complex, the biology cannot occur. But yeah, I was very surprised, but at the same time, I'm very happy that I'm sitting at this end because I'm convinced that there are processes which are in biology as well or in living organism can occur by quantum processes, if you can manipulate that.
Yeah.
But still, it's a long way to go. It's just basically started, or very few researches are going on because basically we don't have equipments or instruments. It's not only us. Basically, in the world, there are very few equipments that can do that or read that, like quantum processes, because basically quantum has to be done at 0 degree or sub-zero degree temperature. And basically at that temperature, biology ceases to occur, So it's a difficult bit. Let's see where researchers takes us. Hopefully with this paper, the paper that I published, some other scientists take over that work and publish something nice or take over something nice.
Yeah, of course. Yes, I would like just to start a very easy question too, because we started very hot speaking about details of things, but I want our general publics, let's say, to understand what we're talking about. And I will give you my probably worst question, because it is how would you define quantum medicine, and how should we care about it? I mean, should we really care about it as a game-changing instrument that we have, and in which perspective? Just to set the base, and then I would like to, talk about your Nature paper. Is it Nature? Is it not Science? It's Nature. I'm not getting it wrong. Yeah, okay, of course. Yeah, no, because all the time. And yeah, because this is the key. The core of the discourse. So yeah.
So I would like to say here, quantum is the next big thing. We can see how much each and every government in different parts of the world is spending if you want to work on quantum technologies for healthcare. It's really a next big thing. And as we know, quantum is the smallest, not like if you're talking about atoms, you have to talk about electrons, protons, and neutrons, which are basically the smallest thing in this world. And people correlate this to like nuclear energy everywhere, like electricity everywhere. But in our body, there are almost every process is governed by electrons, movement of electrons. Basically, if we see about cellular respiration, is how the electron transfer chain happens. And even sometimes the signaling pathways are moved by electrons. So basically, if you can hijack those electrons or redirect those electrons, you can identify whether the cell is diseased or it is a healthy one. So yes, it is a new way of having medicine. No, like if you can read that output, first you can diagnose a disease early, and later on, if you can control those electron transfer or movement, you can have basically a medicine, no, like treat diseases or diagnose or therapies or something like that. So that is what I envisage in future. That would be my future research to somehow, like measure first of all those quantum processes in the living cells, especially first of all, if we can modulate those activities, right?
Yeah. Ivano, do you have any observations?
I wanted to ask more specifically if there is like some specific processes or effects that if you take out the quantum part of it, they just don't appear at all. Like, so let me put it in simpler terms. Where do we see the relevant quantum effects at this kind of scale in these kinds of systems? Mm-hmm. Because as you said yourself, right? Mm-hmm. Usually, to get relevant quantum phenomena, you kind of need to go either very small or very cold or something like that.
So basically, if we talk about biology, it's mostly the enzymes, mostly. If you see about, for example, I'll give you an example of carbonic anhydrase, cytochromes, and even if you see in plant cells, you have, but it's not an enzyme. Basically, if you see about chloroplast, how the electron, like basically how you convert light into energy is basically a role of electron, how it goes to the center, the reaction center, and changes light to energy. This is all the quantum processes. And basically, if you remove that process, which is actually the most essential thing in the life, the photosynthesis, you will not have that processes. In biology, let's say if you have a electron, which is not completely well understood. So let's say electron transfer chain to generate energy in mitochondria. If you seize that process, how the electron transfer that far, which is basically can occur due to tunneling, quantum tunneling, you will stop to see that processes. But still a lot of needs to be discovered because we need proper controls, and so unless we discover those controls, where you don't have electron transfer process, you cannot say for certain that this process will cease to exist.
So somehow it seems plausible that quantum tunneling plays an important role in this story.
I mean, if we see quantum tunneling, as I mentioned, like before, photosynthesis, one of the thing is there. I mean, it's one of the biggest things that I've seen. It's more quantum coherence, basically, but it is a part of quantum biology. But in enzymes, it do occur. For example, in cytochromes, because the distance that an electron travels in an enzyme is much more than the limit that has been shown in theory, like 3 nanometers, 4 nanometers. Compared 3 nanometers, electron traveling is far-fetched limit. Is like you're asking a lot, maybe 300 times higher the distance that they can travel, which is never been reported earlier. But some of the studies nowadays is basically one of the group in Harvard that they have discovered that in basically the enzymes can have these kind of transport quantum tunneling effect, and probably we are the second one. So we basically were the first one to introduce in biology, but we kind of got the same, like in cytochromes, electron tunneling can happen.
I have a question for, people not really familiar with the accurate definition of quantum tunneling. Can we define it and describe it from a very simple point of view?
Okay. So I'll try to define it the way I define here in the university, because I'm from pharmacy division and nobody knows about quantum. So basically, if you think about a hill, a mountain, And if you want to throw a stone the other side of the hill, you need to have enough energy to throw that stone on the other side of the hill. If you don't have that kinetic energy, the stone will fall back. But quantum physics, according to the quantum physics, that stone can still exist on the other side of the hill, because quantum physics sees this stone as a subatomic particle or like a dual wave particle. So a wave can travel to the other side of that. There is a tiny bit probability, because it's a probabilistic. Quantum physics is all probabilistic. So there are some tiny chances that that stone can exist on the other side, even if it doesn't have that enough energy. So the process of that the stone, or you can say the particle, can exist on the other side is called as basically tunneling. That electron or a quantum, that is proton or anything, can exist on the other side of the hill that's crossing the barrier, which is really enough height or which has a lot of energy.
So basically, putting it like this, if I understood correctly, what enzymes do is basically enhancing the probability of reaction, not only by the theory of contact and the lowering of the G energy and everything, but also at the nanoscopic and subatomic level to increase the probability of an unlikely transition.
There could be a possibility of that, 100%. Yeah.
Yeah. This is an interesting thing. There should be, maybe it is now, I don't know, should be taught in the very basic biochemistry courses, because now that we know that is like, it could be something like that going on, it is a completely different perspective in the end. So it might be so useful in the end to obtain a complete vision. I say it just because, for example, I am an electrophysiologist and I usually do, patch clamp, those kind of things. And when I explain to people coming from a general biology background, some details of the very basics of electrical properties of the cells, there is always some kind of perception of these features as something that comes side to what a cell is. But actually, electrical features of cells are wherever in cells and are key. And you know it. I mean, we know it well as of our fields. And it is something that should not be forgot just because it is, as a research field, something of niche. And the same is true for considering always the importance of quantum properties of molecules, also at the level of biology, because maybe it is something difficult to know in a definitive way, but it is important to have it as a notion, as something to think about when you're thinking about something new. No?
Yeah, I agree.
Yeah. So I wanted to point out a couple of things. One is that we're receiving likes, which is very nice. And the second thing is that Andrea asked a question in the chat.
Yes, sorry, me too, because I was talking to you. Yeah.
And it's about essentially the difference between quantum biology and quantum chemistry, right? Or, people call it physical chemistry, theoretical chemistry, computational chemistry, whatever. I guess the basic point of the question is, in this kind of chemistry, people model quantitatively properties of molecules all the time. Right? That's exactly what it is. And so Andrea is asking, what's the difference between the sort of quantum biology approach and the quantum chemistry approach when it comes to enzymes?
So I have a very similar view than Jamel Khalili. What he says, like quantum physics, and then you have chemistry, organic chemistry. From organic chemistry, if you make it more complex, you have molecular biology, and from molecular biology, you have life. So they all are interconnected to quantum physics. So basically, if we see about chemistry or quantum chemistry, you know about the breaking of bonds, and then you have electron spins or electron orbitals there. Basically, they are all governed by quantum physics, because if I go too much into detail about quantum physics, Ivano, if you can support me in that, but the electron spin affects a lot of things. So if you have two different spin of electrons, there has to be, like on the other side. And basically, a molecule can form a bond only if they have opposite spin. But in quantum biology, because they can also form bond when you have similar spin, and because that is what because they can form more and more molecules. So that comes into quantum chemistry. Quantum biology is not that much explored yet. It's very few examples like photosynthesis, some DNA repair examples, but a lot to be studied. But quantum chemistry on the other side is fully supported by quantum physics phenomena, especially
Yeah.
about the Pauli exclusion rules, or if you go to the quantum numbers, no spin quantum numbers and this. So it's fully supported by quantum physics.
Yeah, that's why I think he was asking the question, because Andrea is also a theorist, right? He deals with models.
Okay.
He makes models. So I think he was wondering, as I am, if there's like how the modeling is done in this context. If you want to keep things at the level of complexity level of biology, right?
Mm-hmm.
Is there like any approaches that have been that are being developed right now?
I mean, modeling side, I think there haven't been as much as models for protein side, for especially for enzymes. There have been some modeling done. Mathematical modeling has been done, but I'm not very sure about the modeling, how they have done it, because that's not my expertise in that side, is not on the modeling. But I have seen some people, I have read some papers where they have done the modeling as well.
I see. So you do more, the experiments, measurements, and stuff like that.
Yes, I mean, considering my background, we are more towards experimental side. We would like to have more experimental evidences, and I'm sure general public would like to see more experimental when it comes to biology. We need experiments, because basically theory is very important to support experimental data, but when it comes to applying it to humans, let's say it's the medicine, we need more experimental data as well.
Yeah, no, I completely agree. I was just sort of interpreting.
Yeah.
Yeah, thank you, very good. And in fact, in your paper, I do see a lot of measurements and graphs and numbers.
Yeah.
The kind of things that are done in experiments. So very good.
Yeah.
Yeah. Now I think that's the right time to introduce the story of your paper. I mean, the title of your paper is super telling, because it is, let's talk about, sorry. I don't know what happened to my Adobe Acrobat. It closed while I was reading. Anyway, I'm trying to retrieve it. Yeah. Wireless. So first word, very important, wireless. So using something that's wireless, but electrical molecular quantum signaling, which is an electrical signaling related to the quantum properties of some molecules, and it is all done wireless, and it is already super crazy like that.
Yeah.
So wireless electrical molecular quantum signaling for cancer cell apoptosis. And here comes the super wow, because this means that you found a way to kill cancer cells, and specifically to induce apoptosis in cancer cells, specifically by inducing the self-death process through an electrical simulation that exploited quantum properties of macromolecules. So let's talk a little bit and just describe the story of the research as you prefer, and then we will ask you the details about it. Just tell your story, because it's a very interesting story.
So basically, this idea came from two approaches. First of all, I would like to introduce about a treatment that is generally used in market at the moment for glioblastoma, which is a brain tumor, which is called as tumor treating fields. Basically, in that, what happens is patient wears a patch, those who have brain tumors, and that patch delivers alternating current electric fields. And these alternating electric fields stop the cancer cells to divide. Compared to chemotherapy, it is really good. It gives a 9-month benefit to brain tumor patients, which is really good because it's a very aggressive kind of tumor. Sadly, the patients die within a year or two, but this increases the life of the patient by 9 months. So when I joined as a research fellow, I realized that the electrode size that the patient wears are huge. They are millimeter size, but cancer cell is a micrometer size. So you have that resolution gap. You're using a really big macro size electrode and the cells are actually very small. So the idea was to communicate with cells using electricity. Can we communicate with the cells? No, like if we use electricity. And the second idea was from electrochemistry, which is a big mystery, is how, if we can use this electric field to cause redox reactions within the cell. So merging both of them, we thought that why don't we use nanoelectrodes, which are literally nano size, which are a billionth time of a meter. And these nanoparticles can go inside the cells, cancer cells specifically. And when you apply electric field, these nanoparticles or whatever substance over the nanoparticles we attach can have redox reactions on it using the electric field. And at the same time, we are not touching the cells or like putting actually the electrodes to the cell. So it is wireless. So you're wirelessly delivering electric fields. You're causing redox reaction inside the cells. And using these redox reactions, you can kind of instructing the cancer cells to die or initiate the cell dying process. That was the idea. And the idea came in 2018. It took us 4 or 5 years to develop that idea, work through it, every process. But yeah,
You were very fast.
Yes, indeed.
Considering the fact that you've done a lot of work and it was completely pioneer, you were very fast. I mean, for such a paper, at least you need 4 years. So considering publication, 5 is the minimum. So yeah, I mean, my congratulations, of course, for that.
But one thing I should mention here that this is still a in vitro study, which is in a 2D layer model. And I'm traveling to Italy probably next week to Palermo to
Wow.
in vivo studies. Because if we want to translate this technology to humans, first thing is to study in animals. And because this is basically first time study we want to do in animals. So we have a really nice collaborator in University of Palermo. And I'm going to go there and perform these same experiments in cancer models in animals, in rats, to see if this works. And if this works in animals, we'll apply for a translational funding to have some human trials if we can.
Yes, yeah.
So the
work in Italy, so.
Yeah, really. Ivano, you were saying?
I was just essentially what you're gonna do in that case is ask, please give us money to shock people.
No, actually, if you see there, the potential that we use to apply this electric field is 0.65 volts per centimeter, which is really less. That is one of the, actually, it is going against the theory because it was always thought that you need kilovolts of potential to communicate with the cells. But this is the first time study where we use voltages which are 0.65 volts, which is extremely low, basically, like a tingling sensation, maybe not a tingling sensation. Because what we use here is alternating current. So that is my hypothesis, and it's a part of future research, is what I believe is alternating currents are much better to go inside and like penetrate to inside the cell membrane, while direct currents have more a capacitive kind of behavior. They just stuck to the membrane of the cells. They don't go inside the cells. That is why you need a really high potential, which is kilovolts or megavolts, to go inside the cells. So this is one of the advantages, that you can use really low voltages to work with it. No?
So maybe there are some characteristic frequencies that you need to probe the cell.
Yes, exactly. That is one of my hypotheses, and I'm looking forward to doing this research, because I have a hypothesis that every cells will have a different resonatic frequency. That means, for example, in my work, I realized that the normal cells are not affected by this process, because basically we believe the frequency that we are applying is just specific to the cancer cells.
Exactly, that was my question. How do you reach the specificity of action?
So this is something as a part of future work that we are doing, is maybe some ion channels at this frequency are targeted. We have some RNA, differential RNA gene expression analysis, where we see only the ion channels which are expressed in cancer cells are targeted when this process is. And another factor is the heterogeneity of cells, basically, because every cell is different, the membrane is different, and membrane acts as a capacitor. If we see as a cell as a bioelectronic circuit, the membrane acts as a capacitor. So cancer cell have a different membrane and a healthy cell have a different membrane. So maybe these fields are penetrating inside the cancer cell, but not the healthy cells. And what I have realized, we are doing these experiments in different kind of cancers as well nowadays. It changes from cancer to cancer. So for glioblastoma, it's 3 megahertz. For other cancer that we are doing, it's somewhere around 1.5 megahertz and kilohertz. So it changes with cancer to cancer, which is something really interesting that we have seen.
Yeah, as for it, both a comment and a question, because I thought that you were trying to obtain specificity of penetration of the nanoparticles that you use, that of course are responsive to electric and magnetic fields and are conjugated with the macromolecular part of it, which is able, as you explained well before, to cause the redox reaction, then in turn produces apoptosis. But the thing is that many people, of course, in the nanotechnology field, functionalize the nanoparticle with specific ligands or receptors to make them able to penetrate only some kind of cells and not other. So I thought that you were planning to do something like this, but probably there's not even any need to do it.
No, actually, you raised a really good point in terms of delivery.
Yeah.
Because basically, if we have to inject these nanoparticles systemically, they got to reach to the tumor site first.
Yes.
So basically, we use glioblastoma because in glioblastoma, a patient has to go through surgery because the tumor is in the brain. You have to remove through surgery. So what we designed is during the surgery, when the doctors remove the tumors, we apply this as a spray. So we have a pharmacy division who's developing this spray. So we want to spray at the tumor area, close the wound, and then apply electric field.
Yeah.
But at the same time, we are collaborating with research partners here in the UK. They have a specific kind of bubbles, nanobubbles, which based on the viral vector, which typically goes to the targeted site. So we are trying to introduce these nano antenna that we say inside that bubble and deliver into patients. Yeah, so to answer the question, the targeting is really important. That is something we want to improve in future. That is why we are open to collaborations. That is the beauty of science, that you can collaborate to people, with different expertise, meet them and work together to develop a product or save human lives if we can.
Yeah. Yeah, and as for the variety and the diversity of electrical properties of cancers, it is something that is really interesting, and it's something I became aware only during my master, because a collaborator of my supervisor in Firenze works on this specifically, also potassium especially, but not only, but usually potassium currents, alterations in the common types of cancer, for example, epithelial cancer in the lung. And I was super really, I was not even believing in the first place that it was so important. It was strange to me to think about channels as something important in tumors, but actually they are a lot, because also the metastatic phenotype and migration is very much affected by these properties. And I didn't really expect this to produce a difference, but it was, and it was really a discovery for my personal knowledge, because I knew nothing at the time. I was a young student, but now that I realize more and more that this is something important in the variability and in the phenotype of cancer. So the electrical properties, and also the properties that can be modulated by electrical approaches, which is the other side of it. And I'm super happy to hear about it. I'm really happy to hear about it, because this means that this is going to be something that will be studied more and more in the future. I've always had the temptation to do it sooner or later, because so far it's been such a small field, but now maybe.
There are more and more research and interest in this field.
Yeah.
It is very important because cancer is a very complex disease.
Of course.
We have seen the classical way of targeting cancer, no chemotherapy, radiation treatment. We have to look for alternatives. We have to look for other kind of treatments. And ion channels have shown a really promising approach to see, because it's reported that ion channel expression changes with, like there is a difference in expression in cancer versus healthy cells. So that, I think, one of the way moving forward, to target ion channels.
Yeah. Sorry, I've had a problem with connection, probably. Did you see me freeze? No?
No.
Not really. I didn't really notice, but
No, perfect, because I had problems in receiving the audio, but it went away very soon. Okay.
so much.
Ivano, this is your turn, the physical questions.
Well, so I'm not sure it is a physics question per se, but I am wondering. So you explained somehow how you managed to target cancer cells specifically, and then you also explained that it looks like different types of cancer cells respond to different frequency ranges and so on. And so perhaps, in the future, one could hope to build sort of a dictionary, right? Like, you want to kill this thing, you probe at this frequency. That's awesome. But what I'm curious about at this point is, like, you say you can induce essentially death. You can instruct these cells to die. But how specific does the stimulus have to be in order to induce precisely that response and not something else, right?
Okay.
I'm not really sure like how it works. I mean, could you induce some other reaction that causes the cells to do something else?
So that's what we have done. We have plenty of controls. Now, when we do experiments, we have controls with the normal cells, and we apply electric field to see if there are changes in any kind of, like we have done plenty of toxicity studies to see if just the electric field causes any change in any other normal redox processes, because there are plenty of normal redox processes happening in the cells. We have checked that using molecular biology techniques, and basically as a broader response, whether the cells are stressed or dying or not, which we haven't seen it. So we believe, and also we did like gene expression analysis, which is basically very molecular level. We haven't seen any effects. So we believe that it is much more targeted and only happen when we have the bio-nano antennas in the cells. But having said that, we still need to look too much. We need more and more research to see what specific frequency range that we can use. Because currently we have no like bit gaps between the frequency range that we use, because we need more research funding in order to have more and more people coming and work with us so that we can have more and more research done. So currently, let's say we are using 2 megahertz, 2.5 megahertz, 3 megahertz, but we went to narrow down the range to exactly nail which particular frequency. Let's say 2.15 megahertz.
Yeah, that's exactly what I wanted to know.
It's a
The
lot of
level of
that needs to be done in that way. It's very time consuming process, and yeah, we are looking to hire new people, those who can work with us in these kind of fields. No, so
yeah,
yeah.
imagine the whole machinery is very involved. I mean, you have lots of people, lots of stuff.
Yeah, and
Needs to be
especially when
taken
the
care of.
biology gets involved, it becomes much more complicated. You have to be extra careful, and it requires really a lot of time to replicate the experiments, repeat the experiments, and everything. And yeah, that is why we want more and more people coming to us, and we can work together, make
That's
this.
awesome.
Yeah.
So you mentioned you have collaborators in Palermo, but
Yes.
other than that, is there like what are the main hubs of these kinds of research?
So basically, quantum. There is a very big quantum center here in University of Surrey in the UK, where basically Jim L. Khalili and John Joe McFadden, they are there, which I consider currently they are the pioneers of quantum biology, the one who proposed them. For sure, Nottingham is leading in quantum medicine when it comes to applied, because we are the first one to show that it can actually be applied to healthcare. So by far we are leading that. But if you talk about fundamentals, it can be Berkeley in the US, and there are plenty of groups in the Caltech in California, Los Angeles. They're working on it. And yes, so it is like that, but quite well dispersed within the Europe and in the US. And there are plenty of groups working, but not quite in the healthcare side.
Yeah.
But yeah, to be honest, we needed a whole team. So if you'll read my paper, we have me as a person who can do materials plus cancer, my PI, Frankie Rawson, who's electrochemist. We had a clinician there in that paper because he was the one who gave us the cells from the patients. Because it is very important to have the real cells coming out from the patients. We have physicists who helped us with the modeling, quantum tunneling, no modeling. They were the one who helped us how to change things, the barrier length and all these kind of things. We have cancer biologists. We have chemists, David Amabilino, who's really renowned and very famous chemist. So it's a mixed team effort. It's like you have biologists, physicists, chemists, and that is what it makes science fun.
Indeed, there is quite a lot of authors. But to be fair, the paper is 62 pages long. So
yeah, it's always been a teamwork, to be honest. I mean, there are some people. There's one author called Eloise there. She's undergraduate student.
Oh, that's fantastic. It's a great opportunity.
Exactly. So during the COVID, she worked with me, and she helped me to analyze data, and she's there. We acknowledge the contribution of each and every individual. So it makes me feel really good that the undergraduate participated. It's really lovely.
Yeah. I mean, as an undergraduate having the nature, wow.
Yeah, we're talking the big dogs of academic journals here. In my field, you don't even see them with a telescope.
Yeah.
They will never accept that kind of stuff, but they do accept this. That's awesome. Yeah. So what else could we say about this? I mean, we gave an overview, but is there any highlights?
Yeah, I have a question. You were speaking. Your previous question, Ivana, was about how are you sure that you are producing only apoptosis and damage to cancer cells and not something else? And the answer is that basically the way the particles were engineered was such that the activation of a certain redox pathway led to the typical signaling of mitochondrial dysfunction that leads to signaling of apoptosis and then actual apoptosis. But the thing is that could we, in theory, do the reverse. So could we cure excessive oxidative stress, for example, in the early phases of Alzheimer's and Parkinson diseases in neurons by this? Because in theory, if it could be, this might be a really curative approach instead of curing the symptoms, just treating.
That's wonderful questions actually. So this is one of the things that we have envisaged. It's like to use this approach in reverse.
Yeah.
Can
Okay.
we control oxidative, like stress, the reactive oxygen species generation, and they are involved everywhere. Like not only neurodegenerative diseases, basically all the disease, almost everything. So that is one of the things that we are looking forward to. Now it's like really broad applications. And after publishing that paper, we got a lot of interest from people worldwide.
Yeah.
Suggested us, could you send a sample so we can, like try different kind of enzymes, try to control this. So currently I'm doing that, just trying to send samples to different parts of the world so that they can actually basically try these samples on other diseases, as you mentioned. So what you mentioned is just watch out the space, probably within a year, we'll have an answer to that as well.
Yeah, okay. Well, thank you. This is really promising and something that's happening through an approach which is not really well represented in the therapy and translational research in general, because usually if you speak of translational research, most of the times you're speaking about drugs, so molecule development. And in this case, you're using molecules as a piece of a technology which is more similar to deep brain stimulation. So it's something really unique, and actually it might be very broad in applications, as you said, just because it is flexible. So yeah, I mean, let's hope that it will work perfectly. So
hopefully, fingers crossed.
yeah.
And one of the biggest advantage of this therapy is targeting.
Yeah.
Because basically now what FDA or any drug supplier wants to know, if the patient is safe, and what are the advantages over the current treatments.
Yeah.
So we have compared this treatment with the tumor treating field, which by itself is 1.5 or 2 times better than the chemotherapy. So tumor treating field is wonderful. The Novocure, the company who owns the right, they are 8 billion company. But compared to their treatment, we achieve the same therapeutic level in 3 less. So let's say there kill 50% of cancer cells in 3 days. We achieve that response in 12 hours. So basically, it is quite much more efficient than what we got here. What we need further more studies, as I mentioned earlier, animal studies are really important to see the survival benefits. If it works out like that, we really believe that we can transform this or literally translate this to humans.
That would be revolutionary.
Indeed.
Wow. Yeah, I mean, it kind of reminds me in spirit, right, to when people started using particle accelerators in health.
Yeah.
It's a new approach that has a completely new outlook. Very, very interesting. And indeed, so you mentioned that it's quite flexible, right, in principle. But do you already see, let's say, clear generalizations? I mean, away from cancer-induced death in cancer cells, what other kinds of relevant diseases or things could be treated this way? That perhaps naively one could say, oh, these are completely different things, and then maybe you can use this approach to deal with them.
So basically, I think any disease which is caused by electrical activities can be treated. My first and foremost interest in future would be to treat cardiac diseases. Say heart attacks, because this is the maximum number of lives are taken by heart attacks in the world. And basically, if we see, it's a change in electrical activity of the cells that causes the cells. Now, like even the heart attacks have been now known that there's a change in electrical activities. So I want to learn that. I want to work with people, those who works with cardiac cells. I want to take clinicians, identify the gaps that we can use with these research to basically, like cause the, let's say, muscles, cardiac muscles to beat properly. One of the things, if we can use electric fields to beat them properly. Plenty of other things. Alzheimer's, as you mentioned, Julia, before, a lot of neurodegenerative diseases is there. Next thing, one of the thing I also had in mind is antimicrobial resistance.
Yeah, of course.
Antibiotic resistance. If we can use antibiotic resistance, like can we treat antibiotic resistance with these electric fields?
That would be incredible. Like,
would be a game changer, actually. So there's plenty of things. So one of the data, so I think in a couple of months, we are also publishing this one, that we have seen that electric fields was, so some of the cancers have a anti-chemotherapeutic resistance, or like they don't respond to chemotherapeutics. Some cancer cells develop this kind of mutations. When we apply these electric fields, we see that this resistance goes off and the cells die. So we have seen this in cancer cells, but we want to apply this in bacteria and see if we can enhance the effect of antibiotics.
Yeah.
That is the future directions.
Yes.
But research requires funding and people.
it requires funding, people, time, and luck.
Exactly. That time is the most important thing, I would say there.
This is what I wanted to get to, actually, because this program is incredible, but it probably will take like years and years and years.
Mm-hmm.
So what I'm hoping for you to have to carry it out is a permanent position. But
if the number of labs grows, it can be at least midterm aims might be reached within 10 years.
right,
10
exactly.
20, I would guess, because this is actually pioneering research, but the problem is getting the results, not having the technology. Now you already proved it as a proof of principle. So the difficult part has been already done. Theoretically, now it's all work of ends, basically. So I am optimistic, actually. I'm not working on it, but I am optimistic.
No, actually, the university has been kind enough. They are supporting us. Like to fasten the process if we can just translate it as soon as possible.
Of course, yeah.
University of Nottingham is being really supportive in that case.
And I think that university is also willing for patents, isn't it?
So we already have a patent. So first this was a patent, and then only we were able to publish this one. So we already have a patent. We are just adding more and more, like stuff.
Cases and declinations. Yeah, because this has too much, technological potential to be left aside. So you have to publish afterwards, but once it's done, then you can do it. The problem is having the luck of finding the good conditions for each case.
Yeah.
In theory.
Let's not make bioweapons out of this.
That's the problem. Because this is super powerful as a tool, and it cannot be really easily identified because it's nano-based, so it's like anthrax. So it's difficult to detect.
Yeah, that's why I don't want to go to the quantum physics side, which is a little controversial there. And thank God I don't have quantum physics knowledge that much, so I'm fine with it. Yeah.
You will not be kidnapped by some, general, the U.S. Army or something.
Yeah. Yeah.
But it's great. So what is your outlook? Like, how long are you gonna stay in Nottingham?
So I have secured a position in University of Manchester as assistant professor.
Whoa!
Yes. Yeah. So I'll be moving to Manchester next year, early next year, and I will pursue my research there. And obviously, I would like to continue on this part and a different focus than my current PI. Obviously, I would like to focus more, as I mentioned, like antimicrobial resistance, cardiac diseases, and these kind of things. And first of all, for this, I need to do a fundamental research. Can we measure electrical activities of the cell non-invasively? So as Julia mentioned, she does electrophysiology. It's one of the most complicated techniques ever. I tried.
Come on, no, not that much. It's analysis of data, the problem.
It's really difficult patch clamping.
Yeah, sometimes.
I want to develop a technique if we can measure non-invasively, or let's say fluorescently, the electrical activities of the cell.
Yeah.
And can measure this electrical activity. We can use electric fields to modulate these electrical, like cellular electrical activities. And then basically.
Sorry.
You go ahead.
Would you like to use genetically encoded sensors or nano electrosensor instead of usual dyes?
I think I would do some electric field responsive particles.
Yeah.
I have some
instead of dyes,
which I cannot disclose here, which will respond to electric.
No.
Basically, we can like apply electric field and have a easier readout, which could be a fluorescence. And if we get quantified fluorescence to relate to the electrical activities, would be wonderful.
Yeah, indeed. And as of time kinetic, I mean, as of time decay, because the problem of imaging voltage instead of recording voltage with an electron is kinetic. So do you think that having nanoparticles instead of dyes would help with this?
Yes, so the particles, so my PhD is in materials. Generally, I synthesize lanthanide dope particles, which have lovely, very high quantum yield and a long decay time. So I synthesize particles which are slow decay. So basically, if you illuminate them with the iPhone light for 5 seconds, they will keep illuminating for an hour.
Yes.
So the decay is not a problem in them. So I'm planning to use them for these studies.
Yeah.
I wish I could reveal more, but
no problem. I mean, it's just curiosity.
Those like they can replace the voltage sensitive dyes in future.
Because I can say, I mean, we can say it out loud, they are quite shit because they're too slow. I mean, you see only slow signals when you have action potentials that last in the mean 2 milliseconds, and then you have something like tens of milliseconds of time constants, and you're like that, and then, okay, let's say it is the readout, but it's actually not. So it's average. And all the times, that's why I'm not doing imaging, not even of the genetics. I can disclose it now because we are talking about it. Yeah, because I don't like the idea of having the average. I want to have the signal because I feel, old-fashioned because patch clamp was something fashionable in the 90s, but I still like more. So it was like, I don't know, maybe in the future we use your particles and I will be super happy with it.
That is what my group would be based, is like quantum nanomaterials for like future medicine or something like that. That's what I want to phrase my group as in Manchester. That is my vision future.
Well, yeah, good luck in
Thank you.
This because it's always a big jump and it's super beautiful. So
and now, I mean, you will have the stability to plan and carry out this ambitious research program, because it's as we said, it's very far-reaching. I mean, it integrates lots of moving parts from different kinds of expertises. Really neat that you have this opportunity.
Yeah.
Very nice.
Yeah.
I would like to. There is congratulations from Andrea in the chat.
Yeah, I mean, Andrea is basically the mascot today. This time come on, guys, like and subscribe.
Yeah.
You should do, by the way. Do like and subscribe.
Yeah, I always forget to tell it, but I should say it more often.
Anyway, work. I mean, I don't know. I guess it works when you're a full-time YouTuber who does, video a day or something like that. Smash the like button.
Yeah.
Push the ring button, right? So that you can notification. Nice.
Yeah.
Great. Yeah, I mean, as far as I'm concerned, I'm very happy about this this chat, and I learned a lot. And this sounds extremely exciting. So I look forward to, following
Thank you.
happens. I will stay tuned.
Thank you, Ivano. Thank you so much.
Yeah. Also for me, I really would like, I think that if from the audience we don't get any other curiosity or question, we could go to the conclusion. And yeah, to thank Akhil for his kind presence here. And so for all the news and the vibrant perspectives that he disclosed to us. And really, thank you so much, and congratulations, and of course, good luck, because we all need it in research.
Yeah.
And I'm super happy of knowing that young people in the end make it and can change the field of science one way or another, because we really need it for a really high number of reasons. So thank you so much indeed.
Yeah, thank you so much, Ivano. And I really
and also thanks to Domenico for making
thank you to Domenico.
No, like the connection guy, and is one of my very good friend, and I really enjoy his friendship.
Yeah, likewise, and just shout out to Domenico.
Yes.
So yeah. Great.
Thank you so much. And so let's meet again on MeetScience on next appointment, which will be next week. Am I right? Should be, yeah. So thank you so much again, and good night, everyone.
Good night, everyone.
Thank you for having me.
Thank you.
Bye.